In 1961, French microbiologists Francis Jacob and Jacques Monod proposed an Operon model to explain the regulation of gene expression in the prokaryotes. They received a Nobel Prize for this. What is actually Operon? A genetic unit that consists of two or more structural genes that code for the polypeptides, and also a promoter gene that control transcriptional activities. Lactose operon is actually the first control system for the enzyme production workup at the molecular level, describing the control of enzymes that are produced in response to the presence of the sugar lactose in the Golgi cell.
So here I want to explain about the lac operon structure. Okay, so the lac operon can broadly divide into two important regions, that is structural genes and also regulatory genes. Okay, so the regulatory genes divided into a few subregions, which is lac-I gene, which codes for protein and repressor, promoter and operator. Lac-I is located outside of the lac operon. It is codes for repressor protein that can switch off the lac operon by binding to the operator, thus inhibits transcription. Lac-I is stand for inducible because its transcription usually off but can be induced when a specific small molecule, that is inducer, interacts with repressor protein, which is the product of lac-I.
The lac operon found in E. coli consists of three structures: operator, promoter, and structural genes. Promoter acts as a binding site for RNA polymerase to bind to the DNA, while operator determines whether the process of transcription will occur or not. For example, if the operator is off, which means the lac repressor will bind to the operator, it will prevent the RNA polymerase from binding to the promoter, which means there is no transcription. However, if the operator is on, which means lac repressor does not bind to it, so the RNA polymerase can continue the process of transcription.
There are three important genes in the lac operon that are known as lac-ZAC, lac-Y, and lac-A. Lac stands for lactose, the sugar of the genes made the enzymes for. ZAC, Y, A are the alphabet used in the order name of these genes. The genes lac-ZAC codes for enzyme beta-galactosidase. As you know that the sugar lactose is disaccharide, meaning it is made of two simple sugar, glucose and galactose joined by a glycosidic bond. The enzyme beta-galactosidase breaks the bond between glucose and galactose, converting lactose, which is disaccharide, into two simple sugar. The gene lac-Y codes for the protein beta-galactoside permease, which is basically a duplet protein that fits into the cell membrane of the bacteria and allows more lactose to pump into the cell. That's why it is known as beta-galactoside permease, because it is increase the permeability of the cell membrane to beta-galactoside, which is include sugar like lactose. Gene lac-A codes for beta-galactoside acetylase, an enzyme that transfers acetyl group from acetyl-CoA to beta-galactosides.
And now I will explain about the presence of lactose in lac operon. So when lactose is present, allolactose will be formed, which is a type of isomer. Then the allolactose will originally the repressor protein is bind to the operator. But after that, the allolactose will bind to the repressor protein, causing the repressor protein to change shape and unable to bind to the operator anymore. Then, after unable to bind, which means that the RNA polymerase is able to bind to the promoter and transcribe the structural gene. And after transcribing the structural gene, it will form a strand of polycistronic mRNA. Then this strand of RNA is then translated into protein, which is these three types of protein.
So now we look at the first condition of lactose operon, wherein the lactose is absent. Alright, now we have with us here a simplified model of lactose operon. Now the regulatory gene or LAC-I will undergo a transcription process which produces mRNA, and this mRNA will then be translated into the repressor protein. Now this repressor protein can bind to the operator. Operator. So when it binds to the operator, the repressor protein prevents RNA polymerase from binding to the promoter, which causes no transcriptions of LAC-Z, LAC-Y and LAC-A genes. It cannot be transcribed because the repressor protein prevents it. So the structural gene cannot undergo the transcription process which it normally would, which produce an mRNA. So without mRNA, there will be no translation process. Thus, enzymes such as beta-galactosidase, permease, and trans-acetylase will not be synthesized. So when there is no transcription process, no mRNA is produced, which also means no enzyme is produced. That is all. Thank you.